Half a day. Today we're going to look at the phases of meiosis. Now, previously we looked at the process of mitosis, and in mitosis that was instrumental in repair of your body as well as formation of your body through cell reproduction, and mitosis was looking at that process. Now, meiosis is a little bit different. You can sort of think of meiosis as being mitosis times 2. But the key thing is, as mitosis was important for forming the regular cells in your body or the somatic cells, meiosis is important. It will provide for you the gametes, otherwise known as the sperm and egg. And this is really important for our bodies and for species development because it allows for differentiation amongst the genetic qualities of the species. And that's very important evolutionarily for our species to survive. So one of the things to keep in mind when we're looking at meiosis is the chromosome number. So in a typical cell, we refer to the chromosome number as either haploid or diploid. So when we look at the diploid number, we are referring to the somatic cells or the body cells within that particular organism. And that contains a diploid number of chromosomes, which we can abbreviate 2N. In the human, that number is 46. When we talk about gametes, the chromosome number is referred to as being haploid, which can be abbreviated by the letter N. And in humans, that means we have 23 chromosomes. So one chromosome of our 22 autosomes, and then one chromosome that is our sex chromosome, and that determines what gender we will be. When those two gametes, the sperm and the egg, come together, they will form a single zygote, which will have 46 chromosomes. And that is what we consider to be a human regular number of chromosomes. In looking at these pictures that are from our Prentice Hall biology textbook, we can see the processes of meiosis. So we begin here looking at prophase. And in prophase, again, we have pairing of the homologous chromosomes. In metaphase, we have them lining up along the middle. Anaphase, we can see that the homologous chromosomes are pulling apart from one another. And then that will come down here where we have meiosis 2, where we will have prophase 2, then metaphase 2, anaphase 2, and lastly telophase. And if you notice here, in telophase, we end up with 4 daughter cells. And each of those daughter cells is genetically unique. Okay, so when we begin the process of meiosis, we're going to use a very simplified version of a cell with just one pair of chromosomes. So we can assume that this is chromosome number 1 for this particular organism. Now keep in mind, in humans, we have actually 23 pairs, 23 types of chromosomes. So this would happen with a lot more detail in the actual human cell. So originally we obtained 1 chromosome from our mother of type 1 and 1 chromosome from our father of type 1. Now during the interphase, or the replication part of interphase, the mother chromosome will make an exact copy and the father chromosome number 1 will make an exact copy. So now we will end up with double R chromosome material. What happens here is unique for meiosis in that these homologs are going to join together and they are going to make what we call a tetrad. And that is all 4 of these homologous chromosomes, all of the same type, will get together and line up with one another. When these chromosomes line up with one another and they join together during prophase, a very unique thing happens. And the chromosomes, normally that are in the center area here, are going to actually come together, and there will be what is called crossing over that will occur. And with crossing over, you actually get a swapping of what was originally the father DNA and what was originally the mother DNA, is making unique chromosome structures. So if we were to look at them, this chromosome here will be primarily the father's genetic material. This chromosome will be primarily the mother's genetic material. This one will be partially father, partially mother, and this one will also be partially father and partially mother. This allows, again, for more genetic variation within the species, which is very important for the continuation of a species. So if we were to follow those chromosomes as they went through the various phases of meiosis, we would see that at the end we ended up with 4 completely genetically unique cells. This one would be mostly mom, this one mostly dad, and this one would have a combination of those. Now this is just simply looking at it as if there was just one chromosome pair. But like I said before, in humans we have actually 23 pairs. So this even allows for more genetic variation and can explain why sometimes brothers and sisters sharing the same parents actually look very much different from one another. Without going into too much detail, one thing that we want to keep in mind in our gamete formation is oogenesis, or the process of making the human egg, is a little bit different than what we have simplified in this version of meiosis. But one thing to keep in mind is that the female will produce 1 egg as a result of meiosis. However, the male will make 4 genetically unique sperm when his gametes are being formed. For the female, her eggs are actually begin the process of meiosis while she is still in utero, so that's even before she's born, and then they will go into a state of dormancy, and everything will pick up again when she reaches puberty. As far as the male will begin his sperm production when he begins puberty, and that will go throughout his lifetime. However, female will cease her egg production when she reaches menopause, which varies from woman to woman, approximately ages 45 to 55. But the man is usually capable of producing sperm throughout his lifetime after puberty. To compare mitosis versus meiosis, as far as the number of cells produced, mitosis produces 2 cells and meiosis produces 4 cells. In mitosis, the resulting cells are genetically the same. However, in meiosis, they are genetically unique. Mitosis begins and ends with cells that have a 2n number of chromosomes. Meiosis begins with 2n but ends in cells with the n number, or haploid, and 2n is also referred to as diploid. Mitosis is primarily responsible for repairing and making the organism structures itself, and meiosis focuses on reproduction of the species. So why is meiosis important? Well, meiosis is important primarily for genetic diversity. It provides an opportunity for organisms to have offspring with unique genetics. And the hope is that their genetic combinations will allow them to be the fittest for their particular environment and allow for their species to go on and reproduce further. So the more genetic diversity a particular species has, the more likely that it would be able to overcome any types of obstacles that nature throws at it. So genetic diversity is a big plus for species. Another important factor for meiosis, of course, is that it maintains the chromosome number in a species. So by forming N daughter cells, haploid daughter cells, this allows for the number of chromosomes to be the same when the two gametes fuse together. And this provides every generation of that particular type of organism will have the same number of chromosomes. And that allows for these organisms to have the similar types of features that they will carry to make them unique. And that is all I've got for you today for meiosis. All right, thank you.